or a brief presentation of our co-partner. PetBioCell is a medium-sized company located in Germany, dedicated to offer cancer therapy for pets, exclusively cats, dogs, and horses. They are now in the clinical research phase for an approach called dendritic cells therapy. This approach starts with blood collection by the vet. This blood would be then sent to the PetBioCells, to the labs of PetBioCell, where it's first checked and processed to gain monocytes. These monocytes are cultivated with cytokines to differentiate and mature dendritic cells. These cells are sent back to the vet, where they are injected back in the body of the animal, and where they migrate to lymph nodes and interact with T cells to reinitiate an immune reaction of camouflaged cancer cells. PetBioCell reached the Institute for Multiphase Processes to make this process repeated 3 or 4 times, and the aim of this study was to integrate cryopreservation step at this area after cultivating the cell to not repeat the blood collection and to save the cells when thaw them when they are needed. So we developed a protocol for a proof of concept. We worked with 2 concentration of samples, 1 and 5 million cells in 1 milliliter. We used a cryoprotective agent containing 10% dimethyl sulfixate and 90% autologous plasma, using a cooling rate of -1 Kelvin per minute until -80 degrees centigrade, and then moving the samples to liquid nitrogen tanks, where they were preserved for 4 or 8 or 12 weeks in liquid phase. Then they've been thawed at a temperature of 37 Celsius in a water bath, and a washing step followed, where using puffer or in some cases saline solution. In general, we worked with 72 samples. 68 of them were equine and 4 of them were canine. To these I come at the end. Here I want to address an issue that the total count of the cells differed between the animals, so we had to work with the same concentration but sometimes different total volume of samples. So we worked with 2 concentration, as I said above, 1 and 5 million cells in 1 milliliter, but 2 different total volumes also, 0.2 and 1 milliliter. To this issue, I'll come also at the end. First, the results regarding the vitality after thawing. Here are the results illustrated for both concentration groups. 5 million cells in 1 milliliter and 1 million cells in 1 milliliter. Here we see that we had, for a proof of concept, promising results. We can see a slight advantage for the group with 5 million cells in 1 milliliter, like the minimum after 12 weeks was above 78%, while for 1 milliliter it was around 74% vitality after thaw. And the results were more stable across the animals. Regarding quality, fluorescence activated cell sorting has been made using a major histocompatibility complex class 2 to check the ability of antigen expression by the cells, and a minimum threshold of 10% was set by PetBioCell, under which the cells were declared as not useful for the therapy. And we see here that our results showed almost all the time results over the threshold, but in some cases. And here we see also more stable results in the case of 5 million cells in 1 milliliter. To the issue with the total volume, we studied the effect of the total volume of the samples by comparing the results from the first animal. The samples had the same concentration but different total volume. Here is blue for 0.2 milliliter total volume, and orange is for 1 milliliter total volume. Gray is for control, like before freezing, the vitality before freezing. And all the results were compared after 12 weeks. We see a slight advantage for the samples with 0.2 milliliter total volume regard of vitality, but for quality, it was critically under the threshold, as we see here. For the canine samples, we received the samples after the death of the animal, so we decided to, so we didn't get any plasma from the animal, so we decided to experiment on the ability of an ACL or saline solution to substitute plasma in such cases. We used the same protocol as mentioned above, but changing the CPA and a slight difference in the concentration. And we see here the average results, considering vitality and quality. We noticed also promising results. And it was also promising that the protocol used on equine cells could work on canine cells. In general, we worked with PetBioCell on developing a supply chain protocol with an after-thaw vitality of over 74%. We didn't notice any major quantity loss of cells after thawing along the experiment time after 12 weeks in general. For our approach, the total volume, the concentration of 5 million cells in 1 milliliter showed better results regarding quality and vitality, and a total volume of 1 milliliter was critical regarding quality. Our results showed also the tendency of saline solution to be an alternative for autologous plasma in our approach. We are looking forward now to approve or disapprove our results with a larger number of samples, with a more representative results. And we might test on a wider quality test or wider range of quality tests like migration test or apoptosis assays. And for efficiency reasons, we might also experiment on mechanical refrigerators of, at -80 for freezing and preserving. And also we might experiment with trehalose as an additive for the cryoprotective agent and what this might bring for advantages. Thanks for your attention, and I am happy to take your questions. Here are my results again.